Am I audible? Once confirmed before.
Yes, sir.
Yes, sir.
Okay, good. So, good evening, everyone. We have been discussing about restriction endonucleases. We discussed about the cut site. We discussed about different types of sticky ends or blunt ends. And so, if nothing is mentioned here, it is assumed this is 5 prime, and this will be 3 prime. This is 5 prime. So this one is overhang, 5 prime overhang. So if we want to recombine two DNA molecules or mix them up, construct a recombinant DNA, then we'll have to mix like this, and DNA that are compatible, DNA ends that are compatible because of hydrogen bonding, like ATGC ones, they will base pair and form something like this. But this DNA is not continuous, or it is discontinuous because there is a nick on either side. You don't call it a nick now. Say, for example, this is the DNA and the phosphatase bond broke here, phosphate backbone, then you call that a nick. So basically, these are not, say, the 8 hydrogen bonds. That is insufficient to hold these two DNA molecules for too long. So there should be phosphodiester bond formation here, and the enzyme that catalyzes it is ligase. There are two different types of associations when we mix, as in the previous case like this. That is intermolecular association, when the hydrogen bonds or the sticky ends stick together in a way through hydrogen bonds. Then it is intermolecular, it between two different molecules. Intramolecular is within the same molecule. So we discussed about ligation and the enzyme that is used. We discussed about T4 DNA ligase and E. coli DNA ligase. Predominantly, T4 DNA ligase is used in the labs because it is efficient in relatively much more efficient to ligate even the blunt ends. So they both can do ligate sticky ends, but T4 DNA ligase can efficiently ligate even blunt ends, which E. coli DNA ligase cannot do. And I also described one more part when we discuss about different enzyme. I would want you to know about the requirements for each enzyme, the substrates required for the reaction to be carried out, and also the products that will form, right? Substrates here, you will also need substrates are, the DNA should be continuous here to here. The only difference is there is no phosphate backbone. Say, for example, this whole base is not here. Ligase cannot ligate these two, right? That is the substrate for T4 or DNA ligases. Then it also requires to be charged something like by addition of AMP. AMP is then transferred here, and that energy again, the hydroxyl group, the 3' OH, will make an attack and form phosphodiester bond, releasing the AMP. So you should remember those things. So there are some incompatible ends. As long as you get the ends, as I showed you here, like this, they're compatible, they will bind, and then ligase, you add ligase, and it's easy formation of recombinant DNA. But if the ligands are not compatible, then it's easier to make, it's better to make blunt ends. So there are two ways of how you can make blunt ends. The one way is to fill in this gap. This is a 5' overhang. This gap can be filled. Whereas here you have a 3' overhang, and it's not possible to fill this gap. Why is that? Because polymerization always happens from 5' to 3' direction. So in this case, in the filling in this A, in case of A, there is a 3' OH. So DNA polymerase can just add more and more bases. And it is not adding randomly. It is adding based on the template here, right? It will add complementary bases A, T and T, right? So the substrate for DNA polymerase is DNTPs and primer template junction. This is the prime. This can act as a primer, and this is the template. And you just need DNTPs, so DNA polymerase will come and fill it up. It will fill until this end, and that's it. So you will get the free ends. So it is also called, you can say, just extension. Trimming back is, in this case, where there is no possibility of filling up. The easier one is to chop it off. This is 3' overhang, and if this space should be filled, polymerization should have happened this way, but to happen that way, we don't have a 3' OH, so the best way is to cleave it off. And there we can use exonuclease. Majority of the DNA polymerases have exonucleases. There are two. The polymerization activity is always 5' to 3'. And then there are the DNA polymerases also have exonuclease activity. They have 3' to 5' exonuclease activity as well. But there is also DNA polymerase 1, for example, has 5' to 3' exonuclease activity also. DNA polymerase 1 is special in that sense. Anyway, we can use a 3' to 5' exonuclease activity, and it will digest only this part. So when we choose different types of exonucleases, we should ensure that they will only cleave single-strand DNA. They should not have, the chosen exonuclease should not be cleaving this part, right? This is also 3' to 5'. If you're choosing by exonuclease activity, this is the end of it, and it can degrade it. So there are choices how you make. Usually, of course, majority would be they will cleave this single strand part, the hanging, the 3' overhang can be cleaved. And now we have a blunt end, which can be using T4 DNA ligase. It can be ligated to with some other, the blunt end DNA. That is about polishing the DNA. So one of the things that I would like you to think of, possibilities and probabilities when we are discussing about majority of the experiments, how we want to increase the yield and those. Here we are mixing different DNA molecules, and we are expecting that what we want should come. Often when we study these things, we forget about what all other things may come and how they may interfere in our experiment. And then we should also think about how to minimize them. So this slide is about, say, the different possibilities. Try to look at that. We have a vector. Vector is something like a plasmid, which we will discuss further in later. And we have the gene of interest or insert. We have, say, for example, the ends are compatible. Assume that they all have, on all the sides, there is damage one. That means if we add ligase, then there are several possibilities that can happen. One is intramolecular association can happen like this within the insert itself or within the ligase. Those are circular monomers. We can also have linear dimers. Say this one and this end of the vector and the end here, they both ligated, but the others have remained same. So you may have several vectors ligating, or several inserts ligating, or vector and ligate insert ligating like this. They may form linear dimers, or it could be multimers. So here you have a vector insert, you may have another vector, another insert, and so on. There are also possibilities of circular dimers. Okay, and what we actually want is this one. We want one vector ligated with one insert, and that is the recombinant plasmid that we are looking for. You should also think of it like this. These are so many possibilities, and this is one possibility. If any of them forms more, right? More and more vector and insert gets consumed into this. then the probability of recombinant DNA or plasmid forming becomes lesser. I hope you agree with that. If we somehow prevent these from happening, then we can increase the formation of recombinant plasmid, right? So it's a in any place that we are constructing recombinant DNA, like even in genomic libraries, cDNA libraries, we will always try to think of how to increase the probability of recombinant plasmid, how to reduce the probability of those that we don't want, right? So there is also an enzyme called as alkaline phosphatase. Um, phosphatase, the enzyme that removes the phosphate group is called phosphatase, as opposed to kinase. We will learn about polynucleotide kinase somewhere. Kinase is an enzyme that transfers a phosphate group to the substrate, one of the substrates. So alkaline phosphatase will remove the phosphate group. So if we have vector and insert, right, and this is 3' OH, and here is the 5' phosphate group, and when we ligate, it will form a recombinant DNA. Say, for example, if we have treated the vector with with alkaline phosphatase, the phosphate group that is present here will be removed. What will be left is a 5' OH. It should have been 5' phosphate, but phosphate group is removed, and what we are left with OH. And OH will not form a phosphodiester bond, right? You need a phosphate group here for a phosphodiester bond to form. So while this on the upper strand, you may have the ligation, like as shown here, but if there is no phosphate group, ligation will not occur. So we'll discuss more about this when I have in the next class with the pen and all, so it'll be better in that way. Right now, just try to remember the mechanism of what alkaline phosphatase does. And there are linkers. Linkers are just DNA molecules which we can add to the ends. So at this point, I will stop here teaching. Instead, I would want you to answer some questions. Okay, in the next class, of course, we will also learn about linkers, what can be done, and we will use some drawings and notes maybe. Otherwise, we will kill the bus. And then we'll also learn about adapters. And yeah, in today's class, I would want to do some exercises, trying to discuss questions. So I hope you have your paper and pen, and I would want you to answer these questions. So here is a question. You can assume that the fragment is something like this. Okay, fragment is something like this, and I'm writing the restriction size for this is PST. So I will just put it as P. No, 1 second. Okay, BGL2. We'll write just as B. Oh, wait. It will be easier if I take XMA. So this is present on the vector, fragment XMA. And this is the vector which has the restriction site BAMH1. Okay, that's why I had to change it to XMA. So how will you clone the gene? The purpose of this cloning is, the purpose of constructing this recombinant DNA is just to clone the gene of interest there, or the fragment of interest. So what should be there? I have given you the available enzymes to you are ligase, BAMH1, exonuclease, clinofragment. And the vector has BAMH1. What will you do? How will you proceed with making a recombinant DNA? Anybody, as soon as you're done, you please post it on the WhatsApp group. I will try to have a look at it then and there.
Sir, could you repeat the available enzymes alone again, sir? Like BAMH1, exonuclease, clinofragment, and then?
Here it is there, right?
And then you mentioned something. It's like XMA1 is
Okay.
also.
The fragment has the ends of the fragments have XMA gene of restriction site, XMA here. Okay.
Okay, sir. So the available enzymes are those, and the gene of our interest, I mean, the DNA fragment has XMA1 restriction sites, and the vector has the BAMH1.
That's right. Yes.
So we have to find a way to clone it. Okay.
Yes. It's no big deal actually. It's a simple one. If you can finish it, it will be nice. If you're done, please let me know. This is problem number 1. And here is problem number 2. Same thing. In problem number 2, it has PST1. Yes. So what kind of overhangs does BAMH1 release? I think that is the way we are supposed to think. So BAMH1 gives 5' overhangs, right? 5' overhangs. If you want to, this one, the vector has BAMH1. The fragment in problem number 1 has, I said XMA1. Both of them generate 5' overhangs, but they are not compatible. BAMH1 is
Sir, I
GGA.
think we could, like do the endonuclease, use the endonuclease enzymes with the respective ones, like the BAMH1 for the vector and the XM. I don't know. Like, do we have XMA1? Can we use it for the fragment? Like, I'm not sure. If it's available, then we could use both the enzymes, and then like.
Listen. So I have added XMA also here. Okay, I added
Okay.
that right now. So the best way to answer is that we write it on the paper. I practice a concept called thinking on paper, right? We all do maths on paper, right? So if you can visualize and try to put it on paper, take a picture of it and post it on in the rDNA group, it will be beneficial to others also, and we will quickly move away.
Yes, sir. I'll try
Yes.
to think then as better as possible. I'll try to.
Yes, no
Try to
problem.
be-
There are no, throughout this course, you should also understand that there is no wrong question or no wrong answer. There is only audacity and encouragement. Audacity is the ability to, you feel courageous to say what you want, to ask what you want, and to say what you want. I mean, the question answer, that is when it will be, you have to, I think you should practice that. You're already in final year, so it'll be, there is no point of being shy or being passive about it. You practice it now. When you go into the real world, you will feel comfortable about these skills. So feel free, ask your questions. If I know, I will answer immediately. If I don't know, I'll find out and try to answer. Okay, and there is no judgment on my side, and I hope all the friends will also not have any judgment about what anybody asks or not. If you're still shy, please WhatsApp, or you may email or call me, and we can discuss there. So I'm still waiting for a picture of the answer. Okay, these XMA1 and BAMH1 are generating 5' overhangs. So we could actually fill up the gap using Clino fragment. Anybody knows what is Clino fragment? Anybody knows what is Clino fragment?
That fragment of the DNA which is responsible for polymerization.
Yes, the exonucleases subunits are usually deleted, and the remaining part is Clino fragment. It's named after the scientist who did that, I guess. So we can use Clino fragment to fill up the gaps, and then we will get blunt ends. And then the same thing can be done to the vector as well. Then using T4 DNA ligase, we can blunt end ligation can be done between these two. In this case, in question number 2, I was saying it has PST. I said PST, okay. PST1, and here vector has BAMH1. So BAMH1 should be treated with Clino fragment to polish it, to polish the ends, to make them blunt ends. And PST1 gives 3' overhangs. That means we should be able to use exonuclease to cleave off the overhangs and then polish it, and then we can clone these two. Okay. The next question, this is just the primer question. The next one is, I will give you 10 minutes to solve, and you are allowed to discuss among yourselves and try to do it as soon as possible. Clone the fragment from the cloning vector into the expression vector. Don't worry about this vector and other things. Vector here is just the plasmid, and we will discuss about it. These are just practice, and what we are testing here is your ability to use the restriction endonucleases efficiently. How you choose is the question. And okay, into the expression vector, in the appropriate orientation downstream of the promoter with minimum number of steps. Restriction sites on the plasmids are indicated only by the single letter. So here you can identify which one I meant. Clearly indicate the choice of restriction enzymes and explain your choice, justify your choice. That's what it means. So what you're supposed to do is, here is a cloning vector on which this is the gene of interest. You have start codon and you have stop codon. Now, expression, how do we usually write when we say, if you are talking about genes, you must, if you're doing the assignment well, you would understand it better and more. This is the broken arrow indicates the promoter. And this arrow like this indicates an open reading frame. In all the literature, that is how genes are represented. This would be, the arrow here for the open reading frame indicates the direction to be read for the codons. So this will be start. I can't write good. And this is the stop. Okay. So here in the expression vector, I have already given the promoter direction of the promoter. And so you should be putting the gene of interest in this way, in this orientation. Only then you will have proper mRNA synthesized. So look at the information or the available things that were given to you and explain how would you do that. I hope everybody is trying. Please take a book and practice it. I'm doing this for the in light of CA exams. You will spend more time thinking and very little time writing in the CA exams. So this is an analytical subject. You will be given with scenarios like this, and you are supposed to solve those. So please use these opportunities and discuss and try to attempt. Okay, first, if you are trying to attempt, I'll be more than happy just for trying.
Sir, I think minimal steps, I think we could just use KPN1 as the restriction enzyme.
No, no, I will ask you this. Did you solve it on paper?
Sir, I'm just like a
Please
little
take,
schematically just using it on and just written it there, just with like what are the use of
no,
what is
what
the
you do is take a picture of it and post it on WhatsApp. WhatsApp. Okay. So, I mean, it's also a way of sharing different people's thoughts. It will be easier and everybody will get an opportunity to see your answer in writing. And then probably we'll see if there is explanation, somebody may ask, and then you can explain. Okay? Please post it soon.
sure, sir. I mean, it may not be that clear, so I'll just write
Don't
whatever I.
worry, don't worry about clear and beautiful and all. Just whatever you have tried, you can fair it up if required and just post it. Has anybody done anything? So I'm not going to reveal the answer for this. I think you can take a screenshot if required. And if you all submit the answer for this on the on WhatsApp group by 6 today, okay, then I will give you assignment extension for assignment. Okay, is the deal fine?
Yes, sir.
Now you have to inquire and then come to conclusions. Otherwise, You finish this, you upload, you finish this, post it on WhatsApp group, and you upload whatever you have done for the assignment so far, you upload it. And then I will consider extension. How many days do you need for extension? You're asking on the group? The parallel group?
Shall we discuss and get back to you, sir?
No. It's not that much discussion. Maximum I'll give you is 2 days. That too, if you fulfill these the requirements as I asked. I want answer for this question by 6 o'clock today, by everyone on WhatsApp group, and I want the assignment, whatever you have done with PPT or your notes, you make it a PDF and submit it, and then I will give you 2 days extension.
Sir,
Yes.
so in that assignment, even if some of the parts, if it's taken as screenshots from some of the tools used, is that fine, sir? Like I have the notes also, but some of them using the
See,
screen.
right now, I'm not bothered about the validity or how you present in the assignment that you're going to do today, submit today. At the end, when you present your actual work, video, then I will verify for everything. Right now, I'm going to see how much has each worked. That is the only goal I want. I want to know how much people have been working. Is that understood?
Yes, sir.
Yes. Okay, I'll quickly ask a few questions just for fun. Can anybody answer this, or they identify the correct statement? This is just molecular biology trying to recollect some. We have 5 minutes, we'll finish this. Identify the correct statement. Covalently closed circular DNA and linear DNA are of same sizes and double-stranded. Those are the conditions. Linear DNA can bind more ethidium bromide than circular DNA. Circular DNA can bind more ethidium bromide than linear DNA. Both will bind equal amount of ethidium bromide. Ethidium bromide cannot bind to double-stranded DNA as the bases are engaged in hydrogen bonding. So questions like this should make you recollect a lot around this particular question. So can anyone answer? You don't have to post, just say whatever you whatever you can say, and then we'll think.
The linear would be able to bind to more ethidium bromide than the circular one.
Okay, why is that?
Because there are some spatial constraints in the circular DNA for the ethidium bromide to get into it and bind with it, whereas in the linear,
Okay, how does
the both
ethidium bromide bind to DNA?
it intercalates between the two phosphodiester bonds.
What is meant by intercalation? What is meant by intercalation?
It goes and gets sandwiched between the two phosphodiester strands, the backbone of the DNA.
What do you mean by that? Again? Okay, the bases actually stack, right? They have bases are stacked, and ethidium bromide is also a planar structure, similar to that of the bases. So you can think of ethidium bromide stacking between the bases. That is called intercalation. So the interaction is kind of hydrophobic interaction with the planar surfaces of the other nitrogenous bases there. Okay, you can watch the image or something. So next question, Parvati, you should not answer. Somebody else, anybody. Comparison between deoxynucleotides and dideoxynucleotides. Those who have finished genetic engineering should be able to answer. Pick any option.
Sir, it's presence of H at 2 dash position of both deoxy and dideoxy.
I want you to do also one more thing. Is it 2 dash correct or 2 prime correct? You have to find out and let us all know in the WhatsApp group. Okay. To say 2 dash or 2 prime, what is the most correct one? That's what I'm saying. I'm asking you.
2 prime, sir.
No, many people use dash. I was just wondering if dash is it can be used or not. Okay, I just wanted you to find out. Maybe. Okay, what's your answer anyway?
Sir, presence of H at the 2 prime position of both deoxy and dideoxy.
That is the correct one. D. You're saying option D.
Yes, sir.
Anybody else? I will also tell you this: the more active you are during the classes, the more liberal I get. Okay. Otherwise, the distance will be too much, and then we don't know each other, and then the question papers, everything will be. It's not so pleasant for anybody. So the more you speak, the better we can all get. So anybody else wants to try?
Presence of H at 3 prime position of deoxynucleotide, sir.
Which option is that?
Option A.
Option A.
In dideoxynucleotide, in 3 prime OH will be present.
Yes. Okay. Anybody else? What's your name, please? I can't see the screen, so I know Ajit and Parvati.
Sri Sundarajan, sir.
Sundarajan. Okay. Yes. And then fine. Anybody else wants to try? Okay, we are losing time. We have 4 minutes to 5:10, so maybe I won't say the answer. You can all figure out, and I won't let Parvati answer either. This is too simple. This is also simple. Okay, I will leave this here, and if you want to find it out, it's essential that we be Parvati. You're trying. You can try too.
So option D.
Option D.
Yes, sir. Because the deoxy already it has the only hydrogen. That's why it's called deoxy nucleotide. And dideoxy it will have hydrogen both 3 prime and 2 prime regions of the ribose of the deoxyribose.
Yes. If we had ribose, what would it be? So something like this, say O, I can draw well. This will be 1. I'm not drawing the prime. 2, 3, 4. And here we have CH2OH, something like that. That will be this, that's the fifth carbon. So normal ribose would have OH everywhere in both 2 and 3. When you say deoxyribose, we have H in 2 prime position. And dideoxy, it's H in 3 prime position as well. Right? Dideoxy and deoxy, that's the difference. So what is common between deoxy and dideoxy would be, this is common. But are there
There's both H position at both the 2 prime and the 3 prime position. So yes, sir, that's what option D. That's what I was about to say.
Yes. I'm saying is there any other option? Okay, but that's good. I hope you're getting the sense of the type of questions you will have to answer, and these are molecular biology questions. But I hope questions like these are what you should practice more. We will have sufficient practice, and that is how the assignments will come and so on. For that, you need to have a good enough foundation in molecular biology. That is the sole reason why the assignment was given, and it is made video based so that everybody would have understood the concepts and hopefully be able to, in a situation where you can present it well. Okay. So if anybody found an answer, if all should have found answer for this question that is now displayed, and you are supposed to upload it or put it onto WhatsApp link, WhatsApp group. Okay. I'll stop here. If you have any questions or any other things. If you don't have any questions, then we will stop the class now. Okay? Take care and have fun.
Thank you, sir.
Yes. Bye-bye.
Thank you.
Bye, everyone.